Good afternoon, everybody. Welcome to our second session of the Tufts Research and Data Symposium for Food and Nutrition 2021. This is a panel discussion on nutrigenetics, nutrigenomics, and precision nutrition. Before we get started, I just wanted to let you know that this session is being recorded and that if you have any questions or concerns, including any questions for our panelists today, please enter them in the chat box. And now I'm going to hand it off to our moderator, Kenny Westerman, to get started. Thanks.
Great, thanks so much, Lauren. So yeah, I'm excited to talk about all of this for the next hour or so. I will just briefly introduce myself. My name's Kenny Westerman. I am a postdoctoral research fellow at Mass General Hospital in the Broad Institute. And my research focuses in this general area of this particular topic here, in gene-environment interactions and sort of how the environmental exposures are synergistic with genes and influencing health outcomes. And that specifically focuses on nutrition here. I'm excited to have all of our great panelists here. We have José Ordovás, Milena, Jorge, and Paola Sebastiani to talk about this topic, and hopefully we can all have some synergistic perspectives on this particular topic. So today we'll talk about precision nutrition, and specifically, just to give a brief intro here, precision nutrition is a unique topic that goes beyond simply thinking about what we eat. Expanding to when, why, and how we eat, and incorporating information from each individual's unique genome, microbiome, cultural context, and preferences into this approach. The use of big data in this context allows us to look beyond the mean in order to think about individualizing analysis and providing personalized nutrition recommendations. Within that frame, hopefully here we'll be able to discuss the role of specifically nutrigenetics and nutrigenomics in predicting response to diet, and also keeping in mind the pros and cons of this approach when compared to public health and sustainable nutrition approaches. So the format for the next hour or so, I'll allow the panelists each to give a couple minutes of introduction, basically a little bit about their background and how nutrigenomics relates to their work. We'll spend hopefully about half an hour on a Q&A led by myself. That will divide into three rough kinds of larger topics. Let's call it the science, the data and algorithms, and the cultural context. And then finally, we'll have some time in the last 10 minutes or so for some audience questions. So please, the whole time, feel free to enter your questions into the chat box, and we'll hopefully be able to answer them by the end. So let's then get started off. And maybe Jose, do you want to start off to introduce yourself?
Okay, thank you, Kenny. José Ordovás here. My primary appointment is with the HMRC. My secondary appointment is with the Nutrition School. And I landed in the HMRC as a postdoc maybe 40 years ago. I came under the umbrella of the first center director, who was Hamish Monroe. But it happened that my interest during my PhD thesis was in lipoprotein metabolism. So I joined the lipid metabolism lab that was at that point directed by Arnie Seifert. Well, that was at the time that genetics and chronic diseases was getting born. So I took the opportunity that one of the people that initiated the field was at the Children's Hospital, Sotiris Karathonassis, and I joined his team while I was still at the HNRC, and there I was confronted with an interesting situation, is that the geneticists couldn't understand the variability in phenotypes for the same genotype, and at the HNRC, the nutritionists couldn't understand why people are exposed to the same diet and have such different responses. So back in the 80s, I had that moment of eureka, say, oh, maybe they are related. And that was the birth of what we knew at that point, nutrigenetics or nutrigenomics, that then evolved into personalized nutrition, and now driven mostly by NIH in precision nutrition. So what I am doing now is first learning from my previous mistakes. Second, trying to integrate all the different knowledge that we have been generating for decades, including, by the way, the learning that I had from Paola, remember, BU with Adrian Couples many years ago. That was my first introduction to this, PCAs and pseudo artificial intelligence. So those were good old times. Anyway, so we are trying now to integrate all that. We went from the Nutrigenetics to having all the different omics working towards the precision nutrition, and also, for example, learning that it's not only important what we eat, how much we eat, but also when we eat it. So that's the goal that I have now in terms of our research group, and Kenny can speak about that too, as we were lucky to have him there. But yeah, that's what we are doing, integrating the technology, planning how to use real-time biosensors, the artificial intelligence, the machine learning, and let's leave it like this. And we have time to continue with this during the debate. So thank you very much, Kenny.
Great. Thanks, Jose. On to you, Milena.
So first of all, actually, so I got into the precision nutrition for the precision medicine avenue. So I finished my medical school in Brazil, and then I came here, and then actually I got my PhD across the street from you guys. I was at the time called Sackler School, and I got my PhD in infectious disease. And when I left there, this was like in the early 2000s. This was the beginning of the omics platforms, right? And I went to work in a system biology company, and this was actually the beginning of using AI to try to understand this massive data. We didn't call at the time, we didn't call those big data yet, but this was the first time that instead of like doing like RT-PCR, excuse me, we were getting the DNA arrays, the microarrays that were coming like 20, 25,000 genes expression. And then we didn't know how to understand that, right? And then fast forward, I spent like my career actually always understanding these new platforms and then how we can actually integrate all of that. So basically, I'm actually more from like the system biology point of view, been working from the patient side. And then Gil Blender, he had worked with me some time before. He started this company called Insight Tracker. And I was always fascinated by the way that he approached nutrition in the same way that we approach the system biology, understanding that we work as a body, that our body has all the different systems, and that all those things work together. And that's what, and to understand and to tell someone what they should eat, when they should eat, we needed to understand how all the systems work together. And then finally, after several years, as we say, like Gil and I like talking, we finally, about a year or so ago, I joined the team as the VP of Science. And today, I lead our data scientist, epidemiologist, nutritionist. We have several RDs from Tufts. And an exercise physiologist. And then what we do today is collecting DNA, activity tracker, lifestyle, and blood biomarkers from users and integrating them to give recommendations of how people can live healthier and better lives. And I'm very excited to be here.
Great, thanks, Milena. And on to you, Paola.
Hi, I am a kind of a new person at Tufts. I moved, I was at the Boston University until the end of May in 2020. I moved to Tufts Medical Center last June, and I am in the Institute for Clinical Research and Health Policy Studies as a primary appointment, I'm also the director of the BIRD, the Biostatistics, Epidemiology and Research Design Center. And I'm building a new center for quantitative methods in data science. And fundamentally, I'm a statistician, a biostatistician. And then why I'm here. The question is, what is my connection here? I have worked for many, many years on human genetics, statistical genetics, but very early on in 2004, when I met José, I was one of the first persons to actually try to use complex network modeling to build predictive risk prediction models using genetic data. And that was very pioneer work and very controversial back then. The idea that one could really use machine learning tools to try to do predictive, back then was called personalized medicine. And then it was, it transitioned, the terminology changed into precision medicine. And I was lucky that I started to work with a study of centenarians in 2005. And that gave me the opportunity to actually do a lot of work in real observational studies that are ongoing over the years, and in which things change, the data collection changes, the form change, what you learn about nutrition changes over time, and the technologies change. So that creates a lot of challenges in terms of integrating data that are really, really major. As the biological problem, I have looked at the genetics of extreme human longevity. So I study centenarians and what they have that make them resilient, but also enable them to live for a long time. We haven't really looked in details at the nutrition effect on extreme human longevity, but we are now moving in this area. And so we are in the phase of collecting data about nutrition, as well as multi-omics data. In addition to genetics, we're collecting proteomics data, gene expression in blood, metabolomic data, the microbiome. And the hypothesis is that eventually we are going to integrate everything and come up with a really good understanding of how these factors interact with each other and allow people to live healthy and for a long time. But this is not without challenges. I hope that we have opportunities to talk about the actual computational challenges that this integration is, posing us.
Great, thanks, Paola. So I think this is quite a great group to be able to discuss some of these issues, and I'm excited to get into the Q&A section now. So I will, as Jose made brief reference to, I should give my disclosure that I have Paola and Jose are both on my thesis committee, and I worked at Inside Tracker for a brief time. So if I start tossing softball questions like how they all got so smart and funny or whatever, then you'll know why. So let's start off here with our first section. Let's call it the science. And Jose, if I can start off with you here, do you mind just giving a brief overview of what we mean by the difference between nutrigenetics, nutrigenomics, and if there's anything you think missing from the initial description of precision nutrition, just sort of how these terms are related, but how they might be different?
Oof. This has been the discussion for the last, I don't know how many years, right? Nutrigenetics, nutrigenomics, and that reflects precisely the fact that when we started to talk about tailoring diets to the individual, the technology that we were using was first, the single gene approach, and then we move into genetic risk scores, we move into GWAS, and so on and so forth. So that reflects that we were kind of putting all our eggs on the same basket, meaning that genetics were responsible for that huge inter-individual variability that we are observing for any intervention study, whether it's with drugs or whether it's with diet or exercise or whatever, right? We are always going to find responders and non-responders. So technologically speaking, or technically speaking, we were able to finally look inside the genes, with a pinhole, it's not that we were able to, like now, looking at the whole genomes, sequencing and so on. No, just 1, 2, 3, maybe 10 SNPs. And from there, we were trying to put people in the right position in terms of the response, whether that's to dietary fat or to glucose or whatever. So then it came people like more Nutrigenetics, other people like Nutrigenomics. In one case, you could just split apart saying, well, in one case, we are talking about how different forms of the genes, just to talk very simply, could affect the response. In the other case is how nutrients interact with genes to regulate the gene expression, right, in response to polyunsaturated fatty acids or whatever, polyphenols, anything that is in our diets. So that's how it started. Then it came the concept of personalized nutrition, because kind of involved more factors. It was not only genetics. And this is what we are living nowadays. Milena, Paola mentioned the key factors. Now we talk about the epigenetics. That's for you, Kenny. Epigenetics is your area. Microbiome, chronobiology, but all that exposome that we know now, it really affects the differential response among individuals. And that has been, it has been very clear in the PREDICT study, that collaboration that involves King's College in London, SOI, Stanford, Harvard, MGH, us, and so on, that instead of just focusing on one single, let's call it biomarkers, like could be an SNP, right? Just look at everything under the sun to see what was responsible of that variability in response, using precisely the concept of machine learning. How that study was able to control so nicely for genetics. Of course, the genetics were done, GWAS and so on, but in addition, those were twins. So immediately you can see, okay, what is the correlation in response between twin 1 and twin 2? Well, when you look at that, you say, what I have been doing for the last 30 years, looking at genes. Well, obviously they have a significant contribution, but not as much as we needed to take that to the clinical arena, and start predicting responses. Because for that, obviously you need to integrate the microbiome. The microbiome is there like a workhorse, identifying responses to different diets or behaviors. But also things like what I mentioned several times, because I really like the chrononutrition. What you ate the day before, what you ate for breakfast, or when you ate breakfast and lunch, are going to add that variability that you observe when you take one single measure from one individual. So that really is not disappointing, because it's opening so many new doors that now we can measure, right? It's not that we say, okay, well, we run against a wall and that's it. We are going to stay here. No, we have the way to penetrate that wall with all the technology that we are using, and especially with the interpretation that we can use of the results of that technology, with what Paola will tell us more about in terms of artificial intelligence and machine learning. Now, obviously, I guess, Paola, I already pointed out that it's not a done deal, that we know everything and we can just start, working on it like it was the panacea, the solution. No, we really have, especially when it comes to nutrition, it is very complex. So we really have to put effort, and probably years, until we can make really good use of all these data that we are accumulating individuals to really say, okay, so we can predict your response to this with 90% accuracy, and because of that, we can tell you that you have to eat X, Y, or Z, and you have to eat it at 8 a.m. and at 12:30, whatever. So that's how I see it. And then the difference between personalized nutrition and precision nutrition. Well, different societies, they want to see one name or another, but I mean, to me, it's the same. But anyway, sometimes people get a little bit too involved with these denominations. But what is true, and we can go back, and I will just shut up because everybody has to talk. But obviously, precision nutrition is here to stay. The name and the concept, because as everybody knows, NIH has decided to put the money into it. So again, going back to something that I didn't see when I was describing myself earlier, when I was talking about, let's call it precision nutrition or gene diet interaction or whatever, back in the 80s, people, I remember looking at me and rolling their eyes like saying, what is this guy talking about, right? And now suddenly it has been, it's the future, right? So I am glad. I mean, times were difficult, but now we see the sun rising. So let's hope that it will not get cloudy. I think I'll stop here.
Yeah, thanks, Jose. And I think a couple of important points for that. One is that this nutrigenetics and genomics distinction, that it sort of cuts both ways. We have genetics affecting the way that we respond, and we have the food that we eat then affecting, let's not say the genetics, but what's going on at the genome level and gene expression. So I think that's one really important piece. And then the other thing you touched on that I'd love to hear Paola and Milena's comments on. Genetics is obviously only one piece of the puzzle. How much do we expect that genetics is explaining? Is that going to be about half of how we're in our inter-individual differences in response? Is that a minority and we should be talking more about microbiome, that sort of thing? So if either of you have thoughts on that.
If I can start, I think to me, there's this hypothesis that I have never had data to test, but I think the importance of genetics depends on when you assess the importance of genetics. Is this at birth? Is this at the age of 10 years, 20 years, 100 years? So that is going to make a difference. So it's difficult to tell, if there is a trade-off with 25% genetics and 75% environment or this type of question. It's a dynamic process. But what I'm observing from the data that I've collected now in the last few years is that while genetic effects are very, very small, when we try to make a big connection between genetics and phenotypes, what we observe, they are very far from each other. And effects when we start looking at metabolomics or proteomics are much, much bigger. So they're easier to detect. And that's in a studies that I have looked at. So I think integrating genetics with products, with additional molecular data will help us to understand how things are. It's maybe the challenge in the past has been to look only genetics, only metabolomic or other things. And so integrating everything is going to help. Of course, the big challenge is how. I think we should be honest that there is a big hype on machine learning, artificial intelligence, but there are a lot of challenges to make these things work. Yeah. The other point I wanted to make a link to Jose is that I think there is a lot of nuisance in measuring the nutrition, assessing nutrition in individuals, because it's a lot of self-reported, based on recollection, on whether people are more or less honest. If you look, for example, at smoking, a lot of responders lie, willing or not, because the psychological is difficult to admit that you are a smoker or that you smoke a lot. But in part, the integration of molecular data like metabolomic or the microbiome would also give us a very good measure, proxy of diet. So it shouldn't just be the end product, but it's maybe a good proxy of what is the nutrition, the pattern of nutrition in people. And I'll leave it like that. Maybe Milena. Something.
Yeah. Yeah. So I think it's funny, and I just want to emphasize here, and especially that if we have a lot of so DNA, it's something that it doesn't change, right? But and then so people have this idea that is static, right? But and then, as Paola was saying, that even though the DNA doesn't change, maybe contribution over time, right, is going to change. And I think it's not only, I think it is about the time too, and I think it's going to be very dependent on what it is that you measure. So I don't think any that would be an answer of like, oh, DNA is 10%, 15, 50% explain, because I think it's really going to depend what you measure, right? And I think we do, like José himself has some publications that show that the contribution of DNA, for depending on which biomarker you are measuring, it is different, right? And we see exactly the same thing in our population. And our population is different, that we tend to have actually very healthy self-selected population. And even there, when we are looking at the biomarkers, we can see a very big change depending on the biomarker, right? Even, for example, if you're looking inside of glucose themselves, if you're looking at fasting glucose, if you're looking at A1C, those is already a different contribution from the DNA. And then things get even more complicated when we started to look at not only at one time point of the biomarker that you were measuring, but over time, right? So, like, for example, I can tell you that when we cross our DNA, the polygenomic DNA scores, right, of the risk of having a high or low LDL. And then actually we see that contribution, exactly as Paola was mentioning, it changes on the person's age. Like if you look at a smaller population, we don't see that effect so much. However, because we know that LDL tend to go up with age, then in a higher age, then when we look at that, we can definitely see a much higher contribution on that DNA. So if we are measuring these two populations, a younger population, an older population, you're going to see different. And then actually another aspect is that we also see the DNA score has an impact on your ability to change that biomarker, right? So it's not only about like where your biomarker is, but it's like how much your lifestyle can change that biomarker, right? So I think the answer is much more complex than that, than just like a number. I think it depends a lot on like what is the specific question that you are asking. Are you looking for your response to a life-changing intervention? Are you looking for changing which side? It's very complex. Like it's not a simple answer.
Absolutely, yeah. And I still have this vivid memory of getting my, towards some of the things you were talking about, getting my 23andMe results back. And I read about my coffee section and it said, you are likely to drink less coffee. And I said, wait, did I read that right? I thought you were supposed to tell me whether I should drink more coffee or should drink less coffee, but no, that's not what they're saying, right? And so I think that's one of the challenges here, as you alluded to, is getting towards making recommendations of not what is my predisposition, but what should I do about it, and how will my response differ from yours when we both start eating a Mediterranean diet or something like that. Yeah.
Well, let me point to something that maybe, Kenny, we can reverse the roles. I mean, you are here, orchestrating everything, but maybe you should give your opinion too, right? And it's that talking about the twins and Milena, obviously, yeah, the genetics are fixed through the life of the individual, but obviously the I don't know if to call it heritability, the express heritability, and we can say that changes with age, right? And when you look at the epigenetics or methylation of twins when they are born, is practically identical. But as they grow up, right, they just start diverging. So yes, it's true, the genetic code is the same, but then you have the methylation that changes, and that can create those differential effects that we see through the lifespan. Obviously, there will be other factors, but that is an obvious one. and where, as Kenny mentioned, that interaction or effect of the nutrients with the genes are going to be modulated by the epigenetic factors.
Yeah, absolutely. All right, so if we can step forward into our second section, obviously this is all very interrelated here, but our second section, let's start talking about, let's say, quote, the data. So Paola, I'm gonna direct this first question to you here, which is that as we've been talking about a little bit, it's hard to measure diet in general. It's very noisy, it might not be accurate. And we have a great abundance of correlations, but nutritional epidemiology at least already gets a lot of criticism for the fact that we might be finding a lot of correlations that are not causative. So let's say we have our great fancy machine learning algorithms. How do we fight the not just doing a better job capturing these same correlations that we already know exist, but doing something that's going to be informative?
I think the advantage of machine learning algorithms is that is the ability to look at multiple things at the same time. So there are very good algorithms that can discover patterns. So patterns that in a standard pairwise relation, looking at gene and their effect on some metabolites, so genes and proteins may be lost. So if one can look at patterns that involve multiple genes, multiple patterns of metabolites and lead to particular diet or patterns of nutrition, these algorithms can be very powerful. Then the issue of causality is not resolved by machine learning algorithms. So that's something that I think we should be a little bit more honest in how we describe what these algorithms can do. We can do very fancy correlation, like and display with these impressive networks, but these are still correlations. And so there is the question of what kind of data collection, experiments, or trials one should run to really move from simple correlation to more causation. And I think I start seeing, for example, I understand there is a big RFA now for precision nutrition, in which the idea is really to test different diets and see how different diets interact with the multi-omics data to and have different type of responses in individuals. So with this type of data and the combination of machine learning algorithms, we would be able to come up with really precision medicine. But at the moment, with especially cross-sectional data, it's going to be very hard. And the other thing, which I think is a major limitation of machine learning algorithms, is that over many years, my experience has been that experts in machine learning don't really talk to epidemiologists. They don't understand each other. And so there is a serious lack of understanding of what is the impact of study design on using a machine learning algorithm. This is a major issue, which is often overlooked. And that can also lead to really strange conclusions or strange predictions, simply because there is a lack of understanding that the data you're using is not from a longitudinal study, but it's a case control study. No? And I think so the algorithms have a lot of promise. They can really help to go from univariate relation to more multidimensional relations. But there is a lot of work to be done, and a different type of data also to be collected.
All right. Anybody else have thoughts on that?
Well, Milena, you want to add, because you are maybe more involved with.
I can talk a little bit. And so actually, I love your point, Paola. That and it's funny that we keep hearing that, but it just is so true, right? I mean, your conclusion is just as good as your data, right? And then the bias in the data collection, if the people who are doing the analysis do not understand the bias of the data collection, their result is just so off, and they don't even understand why, right? So I think it is very, very important if the people, and actually I say that for just, and we probably have a lot of like young people here in this call, And then as you're starting to do your first analysis, spend time looking at your data, understanding what is the bias behind your data collection. Because if you do not understand that, does not matter if you're using the most defensive machine learning or whatever it is, your data is just going to bring that algorithm is just going to bring all the bias, and that's what's going to be it. And then the second thing, I think you're right, Kenny, that in the end, all these algorithms, they bring a lot of correlations and we need to move. And then later, maybe it's going to be about like getting all these correlations and then doing some trials to understand the causations of that. But I think we also need to, I think the whole medical health have to learn more from those dynamic clinical trials that the oncologists are so good at doing it. Right, the ones that they go in and then they started with a drug and then move on to the next one. They keep following out, and I think we, as the whole healthcare system, we need to learn from that, because the whole traditional randomized clinical trial, 1 is too slow, 2 is too expensive, and then 3 also brings all the different kind of bias that are coming there, right? So I think we do need to moving in finding solutions to meet the users, the clients, the patients, whoever you want to call them, where they are, to create something that is much more realistic to their normal environment, not so artificial as a control trial, and being very dynamic so we can actually move faster and cheaper. Because I think the whole old concept of like randomized trials just make us things moving too slow and too expensive. We just can't.
Yeah.
I know that's a pretty big a big thing that I think a lot of people are pushing for, and is a very interesting one, as you mentioned, the adaptive. When we're not interested in testing a chemical, not a drug, not even a food, but a method for choosing a food or something like that, where it's going to be adaptive to how you respond in real time, and probably worth considering. And I mean, maybe Jose, I'll push this towards you as I propose it, but an alternative is to say, let's forget all about prediction. We don't need to know your genome or anything else. Let's just have a couple of health parameters that we care about, feed you a diet, see how that parameter changes, and then that's how we're going to determine, sort of in this nature of the type of trial that Milena's talking about. So how do we think about whether we want to predict or we want to just track?
I mean, Milena, what you were saying, and I think, Kenny, that's what you are pointing to, is the N of 1 trial, right? In which you just keep trying. You may not know too much about what is going on in the individual in terms of their biology, but okay, these biomarkers, they seem to be doing better with this specific modification, and you just follow that. I don't think they are as cheap and simple as some people propose. And I have been looking into that for years, and I see that they are not getting enough traction, and they are very difficult to interpret from the perspective of statistics. There are no good statistics for N of 1 trials. So that's my opinion. But independently of that, and just connecting with the previous question that is still lingering of the data, to me, something that it's difficult to assimilate is that we are talking about precision nutrition with measures that are so imprecise.
Exactly.
And I am not talking, that's Paola, that's what you pointed at. And we are not talking about the genetics. We know how to measure precisely, well, at least quite precisely, or even the epigenetics or metabolomics or whatever, right? But when it comes to what it has been mentioned, the diet, that's a no, that's totally imprecise, that's subjective. And that's why there are also RFAs dedicated to see how metabolomics can contribute to know in a more objective fashion what people are eating. Now, pharmacokinetics is key here because you have to, it depends of what you are doing. It will come the time in which you will be able to have sensors that detect what you are eating. Fine, okay. And measure the responses, even real time. But now, by the time that somebody eats something, and then it could be measured, that specific compound may have disappeared because of the short lifetime or half lifetime of that specific compound. So you measure, oh, this person is not eating this because we don't measure that. And it's because simply it's not there anymore, not because the person didn't eat it. So we have that pharmacokinetic component that is going to be a little bit difficult to deal with for every single marker. So that we have that that in the works, right? And in addition, those of us that have been doing metabolomics, we know that there are X number of compounds that they have identified, targeted metabolomics, but then you have the untargeted metabolomics, and sometimes you do the statistics. Wow, 10 to the minus 20 with this compound that metabolite that we don't have the faintest idea of what is that, right? So obviously identification, characterization of those unknowns is important. And that brings me back to the foods. And we are all reading about the fact that everything that we eat, from a potato to watermelon to whatever, has thousands and thousands of compounds. And we know, the low-hanging fruit, excuse me, fruit, yeah. But we don't know the vast majority of things that could be functional, but we don't have a name for them. So we have to penetrate also in that aspect of nutrition, because otherwise we may, and it happens to us in the past, we point our fingers, this is because of that. Why? Because we know it exists, not because it's responsible for. So it's guilty by association rather by that being the one that triggers the gun. So those are areas that it's going to be an interesting future. But as long as we have funding, it will be a future, yes.
If I can make a comment, I agree, especially the challenge of all the unknowns in metabolomic. So there is all the unknown in metabolomic, in which we can measure a lot and we don't know what we're measuring. And then all the unknown in proteomics, in which we can measure little and we know there is much more, but we cannot measure them. So these are very challenging, very, very challenging things, but very exciting. I think the future is bright.
Yeah, for the young people, don't think that we are just kind of telling you don't continue. It's too difficult. No, it was much more difficult when we started, right? And we were able to go over many of those hurdles. So now you have it easier, So go ahead, keep going.
And that's, I think, to summarize what both of you are saying, that's something I know I came into my thesis project, for example, thinking I want to learn and implement the most advanced statistical methods that I can, and that's going to help us solve the problem. And I think what you're both highlighting is that measurement of diet, measurement of all the compounds in the body, it's these issues that are almost more not methodological in a statistical sense, but sort of the instrumentation we're using and the methods for collecting the data are just as important in moving things forward as using artificial intelligence or something like that. All right, so then just to move on to this last piece here, it's very easy, I think, to get caught up in a discussion about precision nutrition. We're talking about molecules, we're talking about inter-individual response and how we can really optimize diet. I think then it's easy to forget in that conversation that there are all sorts of other things that might govern why we eat what we eat or why we might want to. We could think about sustainability, and we could think about the fact that if access to these algorithms or access to these insights are only available to a certain set of people, that these can exacerbate rather than help health disparities. And so I guess for anyone who's interested in addressing a part of this, how do we think about that? And is precision nutrition in fact at odds with some of these sustainability approaches, or is it part of the way we can actually move there?
Okay. I see that everybody's jumping to answer that quick, easy question, right? Obviously, what we don't want is to have all this work going to a few, right? So first, we have to develop tools that are available for everybody. And we have seen it with every new technology, first a few, and then everybody has it. So this is going to be most probably the same. So and in terms of the sustainability, it's the same thing. At this point, where we are and what everything that we can predict, right, in terms of planetary health, and I mean, what we are suffering with COVID is an example of that. We don't pay attention to our ecosystem, and this is what happens. Sorry. So if we don't work in our system, also that way of being sustainable, and I mean, if let's say that we are able to tell people this is what you should be eating, right? I don't know. In my mind, very simple mind, immediately I think, okay, well, probably there is less waste, right? People are going to be, directly to what they have. They should eat, they like to eat, and they don't just move around, and wasting resources. That may be, again, a very simple. It has to be demonstrated, but that could be something positive that we need to incorporate in our research also. How to deal with the sustainability by changing also the materials, the materials that we use in the preparation of our meals, right? Materials is not the right word, but you understand what I mean.
So actually, I think a lot about that, Kenny, because like myself, like we are in a company, we are we are a private company that people come in here and they pay a lot of money for us, right? But what I truly believe is not only to make myself feel good, but what I also truly believe is that any technology, right? We always need to have some people that can that has to help develop a technology so the rest of the population can help, right? So the way I see now is that the few people that can afford our kind of product now, they are actually helping us creating a platform and creating the data so we can actually understand that. So then we can reduce the cost and then apply for more people, right? And that you can see that very well, for example, about the DNA, right? DNA was so expensive, then now we can run some chips for few $100. We can actually get measure, I do not know, like 800,000 SNPs, right? So then I think that's how I see it. I think that is still like a cutting edge technology today, but I think as we increase the N, and we're just going to get to learn more, and that becomes more accessible for everybody else. And then like Jose, that I think that as we understand more and more what people need and what some people do not need, we are also going to be able to suggest what they should eat, but also to understand what are the supplements that some people should have and then the other people should not have, right? And that becomes part of the sustainability, because we are also like not giving something that for people that they don't need. We're just giving them what exactly what they need. And then once we started to understand those, we can actually, once we understand, I don't think we're going to be completely unique, right? I think it's very good for us to, we like to think of us like as one, right? But in the end, it's going to be like more like a stratification of like, this group of people tend to respond like that. These other people tend to respond like that. And then we are able to create just kind of like supplements or create just kind of like environment that become more sustainable.
Definitely, and I think that definitely makes sense in terms of the way that a smaller set of individuals can help develop a technology for the larger group. With that being said, I think the concern that we might have there is what we've seen happen with, let's say, the genetics field, for example, where, okay, let's develop it in the most convenient sample, and suddenly now we look back 10 years later and it's all Europeans, or it's mostly Europeans. And not only that, but the scores and the genetic insights don't fully transfer, and oh wait, okay, so suddenly by not thinking from the beginning, and of course that's not a particular criticism of any particular way of going about it, but it's just something, you know, worth keeping in mind, of course. And Paola, I don't know if you have any thoughts on you know, you were talking earlier about these biases that exist in our data, but are there any aspects that we should particularly be keeping in mind about the data sets that we use or the algorithms that we use to make sure that these biases aren't getting built into our artificial intelligence models, for example? Paola, you're on mute.
Sorry about that. I think, of course, there is one of the issue in a lot of data that we work with that there is not much diversity. And that's one of the challenges of the many genetic studies. Hopefully things will change, and maybe some subgroups of population that are not highly represented, you know, as we start getting bigger and bigger data set, maybe there will be more opportunities to really extend what we learn and apply them to different racial groups. I think that's a really important issue.
Well, that's the emphasis precisely of the Precision Nutrition Initiative from a program from NIH that is going to focus on diversity. It will be based on all of us, the 1 million cohort, that is a diverse population by itself. And it's not diversity only in terms of ethnicity, it's rural, urban, socioeconomic status, and everything that you think that can create that gap, right? So that is in the works, and it will compensate for many of the things that we have not done in the past. Among other things, because we don't have the resources.
Right. Yeah.
Definitely. All right. Yeah. Thanks so much for these answers here. I'll going to give a couple minutes to a couple questions we have had come in. First, have there been any studies done validating improved health outcomes after following precision nutrition advice?
I may just use 1 minute to answer that. I will not, first, if you are talking about improved health in terms of hard endpoints of disease, no. If you are talking about biomarkers or changes in eating attitude towards the better, right? The answer is yes. But this is for perspective. For retrospective, yes. For example, the PREMIER study, right, that was done with a Mediterranean versus low-fat diet. In that case, it was not a prospective precision nutrition, but it was analyzed hypothesis-driven retrospectively. And then we see that even in people at high risk in their 60s, 70s, and 80s, after 5 years of being on the right diet for, in this case, the genotype, you were able to decrease significantly the number of cardiovascular events. So that tells you something that we are moving, you know, in the right track, on the right track in terms of that. So yes, it has not been prospective, but yes, retrospective.
Okay, so another question here. As professionals in precision nutrition, do you have any suggestions for how to reduce the gap for healthcare professionals who want to apply precision nutrition approaches for their patients?
Well, I have an answer, but it's going to sound like I'm promoting Inside Tracker. So actually, we just launching this whole process that we not only we sell directly to consumers, but we also work a lot with the healthcare people, the healthcare physicians or coaches or anything that they wanted to approach the precision nutrition but to their patients, but they do not know how to do it because there is such a big gap, as the person mentioned here, for them to catch up. So then they just work with us, but that's not a solution. That's not, I mean, completely scalable, right? I think we definitely need to. And actually coming from the physician side, I have to tell you that I think the question here is actually bigger than that. It's the fact that I think physicians do not even fully understand how actually food is the medication. I think physicians is still like a lot of that population still believe that it's better to give a pill than to teach them how to eat well. So I think that it's a much bigger gap there. But I think we need to go back. I agree with this person that we need to go back and then closing this loop, because I think teaching people how to eat well and then teaching them how to eat the right thing for their genotype or for whatever condition that they have, that is such a big impact in a person's health that I think that is almost a crime, the fact that we do not address that properly today.
Yeah. And still the public, the general public,
yes,
has a lot of trust for the health professionals, physicians,
yes,
dietitians, nutritionists. And so we need, in the case of the dietitians and nutritionists, they are educated in nutrition. However, the physicians, they have that gap in their education, right? That they didn't have enough in-depth education. So we have to close that gap, as Milena was saying, and educate them too from the beginning, right? Not just as a remedial course later on. And I think something that is important, given the fact that everybody is has time, and we have time as a scarce treasure, you have to make it simple for them. So the final product is something that can be implemented very easily for the health professional, and then the same applies obviously to the customer, to the final recipient of the information.
Really. So just because we're a little low on time here, maybe we can do this wait, don't tell me style. So maybe like 10 seconds or a couple sentences each, but a question from Elena Nomova to take us out here. If each of you could just mention what maybe something you're most excited about. It could be a data set, it could be a method, etc., but something you're excited about in the field of precision nutrition in the next maybe 5 years.
If I can start, I have to say that what makes me most interested in this field is that in terms of the healthy aging and longevity, nutrition is what has been shown to really be effective. And maybe more than nutrition, I can make a joke and say starvation, so reducing the intake of food, so caloric restriction. But that's the only intervention that has been really shown to have an effect on the lifespan of various species as well as humans. So nutrition is really a key thing that can be even more powerful than medications. Just we need to understand more.
Perfect. Milena? Yeah.
So if I am to go next, I think it's the public perception. I think that is what I'm most excited about in the next few years. I think COVID has a lot of very negative things, but I think there were a couple of things that were positive. I think was that people understood for the first time that they need to keep themselves healthier, right? They need to keep themselves healthy because you do not know when a illness is going to knock in your door. And I think that awareness for people, and also this whole idea that some people get the disease, but some people get very sick, some people don't get very sick. So people are starting to really understand and internalize the concepts of like personalization, that you do not respond the same to the conditions in the same way. That together with the realization that you need to invest in your health because you do not know when something is going to happen to you. So I think having the public awareness and having the public in general open to receive precision nutrition, that's what today for me excites me the most.
Great.
Okay, closing arguments, because we are past our time. To me, is precisely the target of precision nutrition. Is the person, is the individual. So to think that in the future, individuals will be healthy thanks to nutrition. That's the most exciting thing. And that means that people will, let's say, experience that even before they are conceived, although that's not possible, of course. But the precision nutrition will start before conception, and it will with a very healthy longevity, right? So that's what it makes me excited about this.
Great. Well, yeah, so with that, I will thank everybody for coming. Thank you to all of our panelists here, José, Milena, Paola, and thanks to Lauren for organizing this. So everyone have a wonderful afternoon.
Thank you.
Thank you so much.
Thanks, everybody.